But we also learned that the smooth endoplasmic reticulum doesn't only synthesize lipids, but it also helps to detoxify substances. And so I sent out that email about, and I like, I'm a huge like Shark Week fanatic person. And I sent out that email because I remembered from Shark Week watching the special where like orcas were biting the livers out of sharks. And this is why great white sharks are like terrified of orca. And it's because much like other ocean dwelling, like mammals and large organisms like seals and walruses have a ton of lipids, right? They're like notorious for their blubber, which is why they're targeted by sharks and whales and orca. And so it turns out that orca were targeting sharks, like they were finding these great white sharks with no livers, which was really insane. And then it's because their lipids, or because their livers are so high in lipid. And somebody emailed me the same thought that I had when I saw this special was like, how do orca know where the shark liver is? And I have no idea. That's like, yeah, like, how do they know? And I don't know. My mind is blown when I think about this.
Trial and error, I guess, and check.
Yeah, trial and error. I don't know. These orca
bite them all over and figure out where it is.
It's just like bite, and they're like, this is a sweet spot. But like, it serves to show like, you know, the evolution like of orca and sharks throughout time, how that, you know, just like us and viruses have really co-evolved, and orca and sharks have similarly co-evolved. Orca are also porpoises, or something like similar to dolphins. And I'm like a human biologist. Listen to me talk about this. But what that means is that porpoises, like dolphins and orca, are also some of the smartest animals that exist. So I guess I wasn't overly surprised that orca are able to be like anatomically, like that's where the shark liver is and bite it. But it is still just like, what? But another thing that came out about that actually is soon after I remembered about the shark liver being high in fat. I remembered that it was a specific fat, and it turns out there is a specific fat that sharks produce, and it's called squalene. And interestingly enough, I remember that this is a very oxygen-rich fat, and this helps with the buoyancy of sharks. So one of the reasons they can kind of like, help float and be so good through the water. But another thing about that is I remembered, I was like, squalene sounds so familiar. And it turns out that this is a huge controversy right now, squalene, because squalene is actually used in some vaccines as an adjuvant. So if you're not familiar with vaccines or adjuvants, for you to establish immunity via a vaccine, or actually via, getting an infection, you actually need to have some level of inflammation. So a lot of people will say, oh, I got this shot and it's sore, or it hurts, or there's redness or swelling at the site of an injection, and people complain about it, but it's actually very good. You actually want a certain level of controlled minor inflammation, because in order to establish long-term immunity, inflammation is actually very important. And so what some vaccines cannot effectively give you immunity directly, and they need a little help. And they need a little help because they need something to initiate inflammation so that they can generate immunity and that the whole system works. So some vaccines, in order to give you the little inflammation, they include what are called adjuvants. And it turns out that squalene is one of those, but it's not used a whole lot. But one of the reasons it popped up in my news feed yesterday or the day before, whenever, was because there's like 22 vaccines being explored for COVID-19 right now that are potential candidates. And a couple of them do have adjuvants. And I think like one of them is potentially like suggesting the use of squalene for the adjuvant in a potential COVID-19. So obviously people were concerned because we worry about shark conservation is the word I'm trying to say. But if this is the vaccine candidate, what this study was showing is using squalene as an adjuvant would have severe consequences on the shark population. So maybe let's explore like a different adjuvant. So they are. We'll see what happens. From what I understand, it's not like the most likely candidate for the vaccine, but I just thought that was interesting, again, because it's the smooth ER that makes squalene that might be used as an adjuvant that also makes, shark livers targets for humans and orca, and just shark liver apparently is just like amazing. So
is there theoretically a way to harvest that fat without
sharks?
No, without killing them. Which arguably might be less humane, but
well, not necessarily. Well, I mean, it's such a fine thing to talk about this stuff, right? But one of the things that I don't know if you've heard about horseshoe crabs, but horseshoe crabs are really interesting because they actually have a component in their blood. So we use horseshoe crab blood to test whether injectables are contaminated with toxic substances. And so we will collect horseshoe crab blood and we'll test an injectable and say, like if it clots, this is obviously contaminated. If it doesn't, it's okay. But we actually collect horseshoe crabs, we bleed them, we take 1/3 of their blood and then release them. We mark them to make sure that we don't, collect the same one. But it is estimated that about 10 to 30% of horseshoe crabs die from this process. So has that happened like where animals are not killed, but their tissue or substance is used from them, and we hope for the best. That is practiced with horseshoe crabs. So I don't know. My question is, we know we can take a little fragment of DNA and we can insert a gene into that DNA and put it in E. coli, and we can produce the heck out of it in a petri dish. And we do that with human insulin. So we actually took the gene for insulin, put it in E. coli, and a lot of human insulin is actually generated in a lab by E. coli. So that's my question. Like, why can't we just do this in a petri dish? Why can't we just take the gene for squalene? I don't know.
How do you get the E. coli out though? Because isn't that like dangerous?
Well, so it's actually a secreted protein. This would be a secreted lipid. So it's secreted out of the bacterium. So when you purify the insulin, or technically if you were to purify the squalene, you wouldn't have the bacteria anymore. So it's secreting, you could isolate it. However, E. coli does have a toxin, notably the one that we use horseshoe crab blood to detect. So there's a lot of, I don't know. I don't know why we have to use horseshoe crab blood. It is blue. I don't know. That has nothing to do with it, but it's a fascinating fact. So those excellent questions, though.
So all in all, you're telling me that orcas hunt sharks with a porpoise.
How long have you been sitting on that, bud?
A little too long, I'll be honest.
That was really good. That's amazing. Hold on to that forever, and I'm so glad that that's recorded. I love it. So awesome. Well, that was I had to share because, yeah, this is such cool stuff. I'm learning a lot too. All right. So are there any questions before I start in on endosymbiosis? Everybody feeling okay with everything so far? Good. All right. It's a fun week. Well, let's talk about endosymbiosis then, which is also a very fun topic. Pretty cool. All right. So when I looked at the, when I was looking at the discussion board for the muddiest points, when I looked at the comments about endosymbiosis, it seemed like everybody had a pretty good idea of like what we were talking about, right? It was sort of like, this is how cells evolve to have a mitochondria and chloroplast. I get it. But it was more of a question of like conceptually, how this, well, one, mechanistically, like how this happened. And then two, sort of, it's just like a lot to take in. And so I love talking about this because it's a very interesting topic. And also because, you know, I get to emphasize why microbiology is so cool. So thank you. So first off, I want to start with just mentioning that there is a different thing between endosymbiosis and the endosymbiotic or endosymbionic, interchangeable terms, theory. So first off, endosymbiosis is something that occurs in nature. And this occurs in nature regularly, right? So endosymbiosis itself isn't sort of like this one-time amazing thing that happened. Endosymbiotic theory is that sort of like explaining two relevant events that involved endosymbiosis in the evolution of all like multicellular organisms primarily. But endosymbiosis itself is when two organisms have a symbiotic relationship. And a lot of people, when they think about two organisms relying on each other, automatically start to think of like parasites, right? So like parasites need their hosts. But with a parasitic relationship, obviously like this, that is not a good relationship because the parasite is feeding off the host, is like draining the host of its nutrients. That's not a symbiotic relationship. A symbiotic relationship is when two organisms have a relationship that is mutually beneficial. Either mutually beneficial or it just like doesn't have an impact on itself. But symbiotic technically means like working in symbiosis, means kind of working together toward like a better outcome, right? So if you're talking, this symbiotic is another word. I think it has its roots in biology, but it is also very prevalent in the like business world about collaborations. Like a symbiotic relationship would make both companies working together toward a better outcome. So, and that's the same thing here. So like what happens with endosymbiosis specifically is it's a symbiotic relationship, but the endo part of this is referring to the fact that one of these organisms lives inside the other. So it's a symbiotic relationship where one of the organisms lives inside of the other. Again, not parasitic, because both organisms benefit from this relationship. So what endosymbiotic or endosymbionic, whatever, however you learned it is fine. What endosymbiotic theory suggests is that the eukaryotic mitochondria and the eukaryotic chloroplast, or plastids in general, actually evolved from a historical endosymbiotic relationship. So this would be two events, and I'll talk about this in a minute, but this is two events where some like primordial cell, which was very likely archaeal, and I'll talk about that in a minute, performed phagocytosis. So remember we talked about phagocytosis, where the cell produces little pseudopodia, reaches out and pulls it in. So some primordial cell performed phagocytosis of a bacteria, and that bacteria had the capability of turning sugars like glucose into just like gobs and gobs and gobs and gobs of ATP, like tons of ATP. And so what happened is this cell engulfed a bacterium, which is really good at making ATP, and all of a sudden that ATP is shared with the cell that engulfed it. And then also the bacteria is like, this is really nice. Like, I'm producing a lot of ATP for me and the cell that just ate me, but also that cell's like made out of sugar. That cell has a lot of sugar, right? A lot of resources to make ATP and to do that metabolic process with. So this was mutually beneficial for the bacterium, which was engulfed, which now had like safety and protection from the elements, but also had like all of the nutrients it could ever want. And it was also beneficial for the host cell, obviously, that engulfed this bacterium, because instead of just like digesting this bacterium for nutrients, it was like, I'm going to keep this bacteria alive because it's giving me like 38 ATP at a time, which is amazing. So what happened though, is that, and this is again like the theory part of it, is that this relationship was so beneficial that when like the bacteria kind of just decided that it's just like only going to live in this host cell and it never left, right? And the cell that had it was like, this is super beneficial, and you know, the cell that engulfed it didn't really have the like a choice, but when it divided, this organelle like divided with it because it's in the cytoplasm. So it just, the choice it made was to just never digest it. And what happened eventually is like, whereas this bacterium was originally free living, is that eventually it just, there was no purpose for it, you know, that was advantageous to live outside of the host cell. And so it just started like reducing any functions that would cause, that would be related to living outside that cell, right? Like, why would it need a flagella or a cell wall if it's, you know, not living outside of this host cell? So it started losing functions that enabled it to be free living. So I'll go through this a little bit more because there is a lot, this seems very like radical, but there's actually a ton of evidence for this. And I want to go through it. But what we think with the endosymbiotic theory, what we propose, is that the mitochondria was a prokaryote that was engulfed, and this prokaryote was capable of cellular respiration, likely a bacterium. The chloroplast was also very likely a prokaryote capable of photosynthesis. Again, most likely a bacterium. They entered, this is as a parasite. Again, this was like a very beneficial relationship, but that initial event could have been like, oh, I was a parasite, but actually, this is pretty good for me. And then the symbiotic, again, this is based on the fact that this was overall a symbiotic relationship, so mutually beneficial. So what's the evidence for this? So there is actually like a ton of evidence for this. It's so neat. So some of it was presented in the lecture, and some of it we will talk about more in the future. But I do want to clarify, I think one of the terms that was a little confusing from the lecture was autonomous versus semi-autonomous. So what does it mean when a structure is semi-autonomous? And I think it's important to first clarify like what autonomous means. And an autonomous organism would be a free living organism. So for example, like I'm autonomous, right? I'm not, my husband may differ. I'm not a parasite, right? So I'm not relying on another organism for my life. So I am autonomous. I am free living. Each of you is free living. Bacteria are free living. So semi-autonomous, therefore, means not completely free living, but having characteristics of like free living organism. So for example, mitochondria and chloroplasts both have their own DNA, right? So they actually have their own genes. And I think you learned this in the lecture. We talked about mitochondrial DNA. There's also plastid DNA. both mitochondria and chloroplasts have their own DNA and therefore have their own genes. And actually, I don't know if this was mentioned in the lecture, but it is true that both of these also have their own ribosomes. So they can take those genes and they can make them into proteins. We'll kind of focus on that later. But what this means is that they're semi-autonomous because although they can't live outside of the host cell, they don't rely on the host cell to like turn their genes into proteins. They have all of the necessary machinery to like make all of their own structures. So this is unlike something like viruses, right? So we could argue that chloroplasts and plastids are like more alive than viruses because they're more autonomous than viruses. Viruses can't make their own genes into protein. They rely on the host, right? So mitochondria and chloroplasts both have their own genes. They can convert those own genes into proteins. As well as that, mitochondria and chloroplasts are also not reliant on the host cell for reproduction. So mitochondria and chloroplasts, or and I guess plastids in general, both reproduce via a mechanism that's actually very similar to how bacteria reproduce, which is a process called binary fission. You don't need to know the mechanisms of binary fission. All you need to know is it's sort of like just one cell splits in half, right? And then each of those cells splits in half. It's literally binary. Bi means two. Fission means a fissure splitting in two. So it's one cell becoming two. That's binary fission. So mitochondria and chloroplasts both do that. So this is different than like the cell as a whole. Like as your cells go through mitosis, and they do that by duplicating the DNA and then duplicating, the organelles and then dividing. But the cell does actually not control the reproduction of mitochondria. So you can have a cell like, let's say, a neuron. Neurons, lots of your neurons are so slow to divide that you have some neurons that won't like go through reproduction in your lifetime. That's why neurological conditions like neurological damage is so difficult to recover from. It's because neurons don't reproduce as quickly as we would like them to. So you could have a neuron though, and just because the entire neuron is not dividing and making more neurons, in that neuron, chloroplasts are dividing, right? Are not chloroplasts. Oh my gosh, neurons don't have chloroplasts. In that cell, mitochondria are dividing, and they're dividing and dividing and dividing. So you're getting more and more mitochondria, even though the whole cell itself is not dividing. Again, this makes mitochondria and plastids like more alive than viruses. Viruses require the host cell to divide to like make more of them, or a lot of them do at least. So they have their own DNA, they have their own genes, they make their own proteins, they divide themselves, they don't rely on the host cell to divide. However, they can't exist outside of a host cell. Like one, I'm pretty sure they can't really escape, but if they got outside of a host cell, it would just be like, they would probably be destroyed because they don't have a lot of like structural integrity. They rely on the cell's homeostasis, but they really have no purpose outside of a cell. Their main purpose is to like help a cell make ATP. So for a lot of reasons, they could be autonomous and have autonomous characteristics, but at the end of the day, they have like no purpose outside of a cell. So that's why they are semi-autonomous. So what is the support for this theory, which is why I think this is really cool, because again, this is sort of like a pretty radical idea, but there is actually a lot of biological evidence that supports it. One, I noted that both mitochondria and plastids have their own genes and have their own DNA. So your DNA, like eukaryotic DNA, is all linear, meaning like you've probably seen pictures of like chromosomes, they're like X-shaped, but essentially what that means is your DNA has ends. So we talked about that when we talked about like nucleic acids having like a 5 to 3 prime end. And we'll talk more about it later, but they have like distinct ends in eukaryotes. Prokaryotic, so both bacterial and archaeal DNA is circular. So there's no ends, it's like connected end to end. Mitochondrial and plastid DNA is also circular. So structurally, their genomes are similar to prokaryotic organisms. They divide by binary fission, which is very similar to, if not exactly like, how many prokaryotes go through reproduction. And then also several of their genes are very similar to like prokaryotic genes. So they even have some like remnants of probably genes that were relevant when they were free living organisms. So how did this happen? This is just a picture. I like this picture because it's easy to look at, but I actually like hate this right here where it says oxygen breathing bacterium. This bacteria don't have lungs. They can't breathe. What this should say is this is a bacteria that can produce or can undergo cellular respiration, which is taking like carbohydrates like glucose and turning it into ATP. They don't breathe, but they can do cellular respiration. So the hypothesis, well, I guess it's a theory because there's a lot of support for it. The theory is that you have this sort of like original cell, and this says bacteria, but I think a lot of the data suggests that it was actually archaeal. This cell, we'll talk about the nucleus in a minute, but this cell went through some changes, but this cell was able to phagocytose a bacteria that performed cellular respiration. And then that both the host cell and the bacteria were like, this is a really great situation. We're never separating. And then, so that's how we got the modern day mitochondria. So you can see a lot of mitochondria in this cell. This cell, you know, divided, but the mitochondria divided itself many, many times to create that many mitochondria in a cell. So in history, what evolved first was animal cells, because the first acquired organ, like semi-autonomous organelle, was a mitochondria. We know this because animal cells only have a mitochondria, but plant cells have both mitochondria and plastids. So what likely happened is we had this cell, this original cell, and it engulfed or phagocytosed this, what became the mitochondria, and then those cells kind of like went on to live their life as animal cells. But then some of those cells, one of those cells, I guess, went on to further phagocytose a bacterium that could perform photosynthesis, or basically using light energy to generate ATP and actually generate sugars, carbohydrates. And then this cell that had both mitochondria and plastids evolved to modern day plant cells. So this seems like really big events, and I think that's one thing when I talk about endosymbiotic theory, a lot of people are sort of like, I mean, like, how unlikely is that? Like, how fortuitous is this? And the answer is it's actually not that random. So tons and tons of organisms, we have seen endosymbiotic events like continuously. They happen every single day, right? So they're still going on. Like, there are endosymbiotic events that happen every single day. Like, it might have even happened in your body today. Like, who knows? You have phagocytic cells, yeah, right? So it's not a, like, endosymbiosis is not a groundbreaking event. But in our evolutionary history, these in particular were two very obviously impactful endosymbiotic events. So they're not the only endosymbiotic events that have ever happened or happen. It's just that obviously they were really, really impactful. Like one of these led to the mitochondria, which enabled cells to be, to generate a ton more energy. And if you're able to generate that much more energy, you're able to be more complex. So this is what eventually led to more complex cells, more functions, and therefore multicellular organisms. Same thing, with the, like, grabbing up the photosynthesis, that allowed these cells to be independent. Not only could they, now use light to generate sugar, but they had mitochondria to use that sugar and make a ton of ATP. So it allowed these cells to be much more complex. And those were just two really important endosymbiotic events that enabled such drastic complexity. So again, that's what I think is one of the sticking points. You present this theory and everybody's like, oh my God, it's so random. How? It's well, no, this actually happens every day. It's just that these two events that we're talking about were very impactful on development over time. I'm not going to talk a ton about the nucleus because, to be honest, I don't know anything about how the nucleus, I don't know what the nucleus, where the nucleus came from. There are a couple of theories. I do believe the most prevalent is that this ancestral, probably archaeal organism started to have its plasma membrane fold in, and that's what led to, the membrane bound organelles, the endoplasmic reticulum, etc. There's some other suggestions that maybe the nucleus was also an endosymbiotic event, and I don't know. I looked through the literature to try to see what I could find, and just from my understanding, it's not as definitive as the data about mitochondria and chloroplasts is. So it's just, yeah, I don't know the answer with the nucleus. I don't know, which is okay. I do want to also mention something kind of fun about this, is I keep mentioning, one of the big questions with this is what was the original organism that was doing all this engulfing? And one of the original labs, or a lab that kind of is proposing or suggesting an organism for being this organism, I guess, is this lab. I think it's in Norway or Sweden, I can't remember, but they isolated an archaeal organism from a hydrothermal vent, and the vent itself is called Loki's Castle, which I think is really funny. And so because they isolated this archaeal organism from Loki's Castle, they actually gave it the superphylum Asgard. I have this paper here, and I can give it to you if you want, but you can see they gave it all names related to to Asgard, which I thought was so fun. I think there's a Thor, and there's a lot more. But anyway, so I think this is the suggested now living organism that originated, or kind of is existing still, from that original primordial pre-eukaryotic cell, which was archaeal. Archaea have a lot of eukaryotic features, obviously prokaryotic features as well. So one of the big arguments against archaea being this pre-eukaryotic cell is something that we hadn't talked about in class, which is that one of the big differences between eukaryotes and prokaryotes is that eukaryotes have a really, really robust cytoskeleton. So prokaryotes have a cytoskeleton as well. It's just not as robust as eukaryotes. So eukaryotes, in lecture this week, I can't remember, you talked about the cytoskeleton, and you talked about actin, filaments, and some of the other cytoskeletal proteins. And actin is actually really important. What eukaryotic cells do is they use actin to create like the pseudopodia for phagocytosis. So what that would kind of imply is that like without actin, you might not be able to do phagocytosis. And archaea were not really known to have something that could do something like actin. And then you can see here that a study came out that was like, oh, maybe archaea do have something similar enough to actin to where they could do phagocytosis. So I think there was a paper that came out not too long ago that was actually kind of challenging the idea that Loki, I think it's Loki archaea is what the species is, or the genus maybe, but was challenging that this was the original primordial cell that kind of kicked off eukaryotic evolution. I mean, we just don't know definitively. So there's still research going on about it. So if you want to go look at Loki's castle and investigate all the bacteria in Asgard, you can do it. All right. So that is my information about endosymbiosis. So before I move on to talking about cell junctions, which is what I'm planning on talking about next, are there any questions about endosymbiosis or anything like that?
So if the nucleus was like supposedly formed by endosymbiosis, would it be like very similar to the mitochondria, except that it like kind of took over the cell, or?
Yeah, that's sort of, there's some evidence, like the evidence that suggests that the nucleus resulted from an endosymbiotic event is a little bit, again, like less clear, because maybe it was like a parasitic event and then the host cell actually lost its DNA and the parasite, which is now like the nucleus, I guess, ended up being the source of genetic information in the cell. Another, like one thing that supports it though, is that as we will kind of talk about next week, and hopefully it was mentioned in lecture, is that plastids and mitochondria have double membranes. So like the mitochondria has an outer membrane and an inner membrane, and chloroplasts and plastids do too. The nucleus also has a double membrane, implying that perhaps, you know, it was a single membrane, and when it got engulfed, that second membrane is like the vesicle that it was pulled in. But I think the data out there, you will talk about this again in 182, and I think that Professor Chichi has a lot more definitive idea about where the nucleus came from, but every time we talk about it, I don't know. She's pretty sure she thinks she knows what happened. I'm less sure. So make her talk about it though.
All right. I will note that.
Good question. All right. So if there's no questions about endosymbiosis, I'll move on to cell junctions. Somebody the other day yesterday told me that endosymbiosis to them clicked when they thought about the movie Venom. And I haven't seen that movie. So I know that's horrible. I want to see it. I'm obsessed with Tom Hardy, but I have a 4-year-old and I only ever get to watch, you know, Paw Patrol. So my goal is to watch Venom and tell you whether it does support endosymbiosis. So, all right. So we're going to move forward and we're going to talk about cell junctions, which is one of my favorite things. All right. Okay. So cell junctions. I think that when I looked through the comments on the discussion board for cell junctions, I really, again, I think that one of the main points of confusion for cell junctions was that the textbook went into excruciating detail about cell junctions, giving them like tons of very specific terms. But remember, please, please, please focus on the terms and how we're defining these things in the lecture. And the reason I mention that is because, again, the textbook went into excruciating detail, like good stuff, but you don't need to know like the difference between desmosomes and hemidesmosomes for Bio 181. That will be very nice to know, like if you're progressing to anatomy and physiology or something like that. But for Bio 181, we want you to know sort of more the general cell junctions. So these are like the broad categories of cell junctions. Importantly, cell junctions, of which there are like 3 primary types, help basically just connect cells either to each other or to some sort of supportive framework that would enable the building or like, you know, scaffolding of a multicellular organism. So I'll show that in a minute. But again, there are like 3 types. The first is called tight junctions, which basically kind of like snap cells together at near their surface. Desmosomes, which are very, very powerful and basically hold cells together closer to the bottom of a tissue or to underlying tissue. And then we can have another type, which is basically just little tunnels that are between cells. And these are either gap junctions or plasmodesmata. Gap junctions are present in animal cells, and plasmodesmata are present in plant cells. So that's the big difference there. Let's talk about each of these. So first, to sort of orient you, there was some confusion about like looking at tissues and understanding sort of like what is the basement membrane, like what's connective tissue. These are excellent questions. So first off, it's important to know that tissue in general is just lots of cells working together toward a common function. So as I'll talk about in a minute, you have 4 primary tissues: epithelial tissue, muscle tissue, nervous tissue, and connective tissue. And they all work together to sort of like build an organism. So this picture is showing epithelial tissue. And epithelial tissue is used to basically like line and sort of like divide all the spaces in an organism. So because it lines things, I can tell you one really good kind of like area where you have a ton of epithelial tissue is your skin. So a lot of your skin is composed of epithelial tissue. So if you think about your skin, you can't like grab your skin and twist it. Like, well, you can, but when you do it, your skin is not like twisting around your entire arm, right? So if you put your hand on there and twist, it only moves so far. And that's because it's held in place, right? Not only that, but when you put your finger on your finger doesn't just like, your skin doesn't just like move away to accommodate your finger, right? That's because of cell junctions. Cell junctions hold your tissues together and also hold them down. So there's different junctions that do both of those things. But first, let's mention just about directionality, because this is very important. So first off, what you're looking at here is a tissue. This is epithelial tissue. And it does have a top and a bottom. So the top is sort of like with my skin, I'm touching the apical surface, or kind of the uppermost or outermost layer of a tissue. The bottom of a tissue, at least with epithelial tissue, so with my skin, the bottom of my skin is attached to like underlying connective tissue. And that's what makes my skin able to attach to like underlying muscle and underlying bone, etc. So obviously like attaching things is important. You can also think about like connective tissue, and the bottom of things is how like bone, muscle is connected to bone, right? That's here kind of what we're looking at. You can imagine this sort of like the cells of your skin, although that's not what this tissue is, but let's, for the sake of this talk, let's say that these are like the cells of your skin. This would be the surface of your skin, and this would be the like underneath your skin. The basement membrane is a layer of connective tissue that would hold your skin to like underlying muscle and bone. So we can talk about the top and the bottom of a tissue. If you're talking about the top of a tissue, that's called the apical side of that tissue or apical surface of a cell. So when you say apical surface, that just means like toward the top of a tissue. So again, like touching your skin, the surface of your skin, you would be touching the apical surface of the cells that make up your skin. How your cells are attached to underlying tissue is they are basically anchored to that underlying tissue, but obviously that's like the bottom of the cells. So the bottom of cells is called the basal surface of a cell. So this gives us just directionality, but one of the reasons this is important is because in a tissue, we do have different junctions, sort of like in different areas. So for example, tight junctions, which I like to think of tight junctions, I think I mentioned this in the lecture, as sort of like snaps or buttons. But you can notice that tight junctions are located near the apical surface of cells. And this is important because if you have a tissue that's lined with stuff, right? So let's say this was my skin. I don't want to be able to put my finger like through my skin and my cells of my skin just be like, okay, you know, come on in, right? So one of the things that happens with tight junctions is they hold the cells together very tightly at their surface, at the apical surface. And what this means is that those cells are held so tightly together that stuff isn't going to be able to pass between them. So for example, here you see in this tissue, you have the apical surface, and between those cells are held so tightly together that stuff isn't going to be able to pass between them. So that's the purpose of tight junctions. Again, just kind of like holding them together at the top, sort of like this, so that stuff can't pass between. Then you move a little bit deeper, so toward the basal surface of cells, or toward the bottom of the cells, and you have what are called desmosomes. So desmosomes are a lot more like, they're a lot stronger connections compared to tight junctions. Tight junctions are strong too, but so like tight junctions, you can kind of see if you look at them, you have just like a membrane protein. So it's literally like a membrane protein holding two cells together. Desmosomes, if you look at them, you have a lot of protein fibers, and it's sort of like if my fingers were protein fibers, instead of tight junction being like this, desmosomes are like, right? So it's just many more connections, so it's a lot stronger, which is important because one of the big purposes of desmosomes is to hold tissue down, to anchor it. So again, if you like grab your arm and try to twist your skin, it only goes so far because it's held into place. Same thing as well, desmosomes are very prevalent in holding like your like tendons to your bone. So they're very, very important for like making sure that tissue stays where it's supposed to be. And so you will find these on the basal surface of cells, holding them down on connective tissue. So this is again what is going to hold muscle cells to tendon and then tendon to bone, so hold everything together. But also you're going to find some desmosomes between cells as well, because those cells need to stick together, right? Especially in like cardiac tissue, you need the cells in your heart to be very firmly attached to each other, especially if they're going to like work together to contract. Which brings me to the third type of junction. So this is focusing on animal cells, this slide is. So the third type of junction isn't necessarily only, doesn't have the purpose necessarily of like holding cells together or holding them down. Instead, the purpose is to allow cell communication. So these are gap junctions. So gap junctions, you can see here, gap junctions are formed closer to the basal surface of cells, but they're essentially like little tunnels between the cells. And the tunnels themselves are called connexins. So gap junctions are regions made up of several little tunnels formed by proteins called connexins. And these proteins arrange so that they form a little hole like a pore. So I think this is kind of what I talked about in the lecture, like the importance of allowing pores between these cells is that if this cell gets a message or if this cell gets a message, cells communicate via chemical signals in the cytoplasm, which we'll talk about, which we're talking about this week. And so it's important that if you're getting a message in this cell, if that message has to be like a tissue-wide response, that message needs to be unified between all of the cells in the tissue. And that's what those gap junctions allow, right? So you only have so many neurons. You don't want to have like a ton of neurons. And because you want a unified message, so for example, like your muscle cells, like let's say your bicep, is composed of a ton of muscle cells. But you want to make sure that when you want to do this motion, that all of the muscle cells composed in your bicep get the same message. And so really the most kind of like reasonable way to like get a unified message is to have that like one message from one neuron, right? So neuron's like, I want this muscle to move. But you need to make sure that all of the cells in that muscle get the same coordinated message from that one neuron. And how that's going to happen is you have one cell like come in contact with that neuron, but all of the cells in that tissue get the message through gap junctions. So like when this doesn't happen, you'll feel something a little bit weird. So if you've ever had like a twitch in your eye or like a twitch in your muscle, that's like a neuron firing, but it's not the entire tissue responding. Instead, it's just like one of those cells. Like I hate when you get that twitch in your eye, but that's just like one muscle cell for some reason firing. And luckily it's not an entire tissue response, but that's like what a twitch is when you don't have that unified message for whatever reason. There's not something wrong. It's like a common thing. So those are tissues. Gap junctions, again, in animal cells. In plant cells, you have the additional complication of the plant cell wall, which is just like a more rigid structure. The plant cell wall can make communication with the outside environment as well as cell to cell a little bit more complicated. So instead of gap junctions, plant cells have what are called plasmodesmata. So very similar. It's just that instead of being a tunnel that is composed of connexins, cell to cell, it's a pore that's located in the cell wall. Not really exciting there. Maybe it is if you're a plant person. Okay, and this was actually created by a student in class and I wanted to share it, which is a really good table from notes. So just kind of summarizing the different junctions, which I thought was very useful. So I did have a question that I thought was a very cool question. Again, like with those discussion boards, always ask like, you don't have to feel obligated to ask questions out of confusion. If you feel pretty comfortable about it, ask, but you also use them to ask questions out of curiosity. 'Cause I like really, if we can talk about that, it makes my life great. I want to clarify any confusion, but if we have time, I'd love to talk about, you know, stuff that's relevant, that's just like interesting. So I had somebody ask like, okay, I understand tissues. What if I didn't have a tissue? Like, what if I didn't have one of these types of tissues? And the answer is, well, in some respects, so for example, if you didn't have nervous tissue, you wouldn't have a brain or a spinal cord and you wouldn't be alive. If you didn't have muscle tissue, your body wouldn't be able to function very well. Muscles like are not, you can think of muscles as helping movement of large muscles, but your heart is also made out of muscle. Your blood vessels have muscle in them to help like move blood through. your your intestines, all your organs are made of muscle. So if you didn't have muscle tissue, you'd be in very bad shape as well. Epithelial tissue is what makes up all the linings of your body, so obviously very important as well. You would not be able to function without skin. It's very important. And you also wouldn't be able to function if all of your organs didn't have linings. So that brings us to connective tissue. Could you function without connective tissue? And this is actually kind of an interesting question, because the answer is like, not really well, but in some cases, yeah, you could function, but again, like not well. So I want to give a little bit of a warning. I'm going to like transition to the next slide, which is a little bit, it shows pathology, which is like diseased tissue. So if you're grossed out by teeth or bleeding, then maybe just like turn away for a second. So I'm going to show a picture of periodontal disease. Okay, so this brings up what I want to talk about, which is scurvy. So you've probably heard of scurvy when you, like if you watched or have read anything about pirates. And that's because scurvy kind of was associated historically with sailors and pirates, because it actually was quite common disease for people that sailed for long periods of time. And this is because scurvy is a disease characterized by vitamin C deficiency. So importantly, vitamin C is a really important component of connective tissue. In particular, it's an important component of collagen, which if you remember talking about our lectures last week, collagen is one of the really, really important proteins in the extracellular matrix, which is very, very, very important for connective tissue. So vitamin C is important for collagen fibers, and scurvy is a disease that comes about when somebody doesn't get enough vitamin C and therefore has defective connective tissue due to defective collagen or insufficient collagen. And because your connective tissue has so much collagen, what starts to happen are some of the symptoms of scurvy are weakness, because connective tissue is important for holding your muscle cells together, important for like tendons, which hold, you know, muscle to bone, and ligaments, which hold bone to bone. So all of those like things that connect you together. Connective tissue is also really important for building your capillaries, your blood vessels. So you'll see ruptured capillaries, which is why you see bleeding, right? So the vessels that hold the blood are not very structurally intact. And those are some of the earlier symptoms of scurvy, that weakness. The reason in particular that you get bleeding noticeable in the gums is because gums, well, one, they're like very visible. They're a very visible place where your like teeth are held in to your gums. So, and also when you smile, like there they are. But another reason is because your gums are extremely vascular. You'll probably notice this anytime you like haven't flossed your teeth for like what seems like 2 days, and then you floss it and your gums bleed. So it's like they are very, very vascular. There are a lot of capillaries, a lot of blood vessels there. And so there's a lot of connective tissue there. And so when that connective tissue starts to break down, you'll see bleeding. You'll see tooth decay because those capillaries also feed nutrients to the teeth. So if the capillaries are rupturing in the gums, they're not efficiently transferring nutrients to the teeth. And so the teeth start to die. So that's some of the earlier symptoms if scurvy is not corrected. So if this vitamin C deficiency isn't corrected, it can lead to like the larger vessels rupturing, causing systemic issues, abnormal bone growth, etc. So why did this happen so much in sailors, and why is this associated with pirates, is because obviously we get vitamin C from foods that are rich in vitamin C, but the foods that are rich in vitamin C are foods that are very prone to spoilage. So this includes like citrus, like obviously oranges are notorious for vitamin C, but it's also very prevalent in broccoli, cauliflower, peppers, spinach, tomatoes, you know, stuff that would not survive months or years on a boat. And so what would happen is these sailors would go out and they would be relying on like not fresh food sources, and they would develop scurvy because they didn't have vitamin C. I want to mention this, like, I want to talk about this for a second though, because probably when we talk about scurvy, again, you're thinking about like pirates and kind of historical issues with sailing, but scurvy is not, scurvy still exists, and it's actually still a huge worldwide issue in a lot of places where people don't have access to fresh foods that are vitamin C rich. So that's actually a lot of places on this planet, unfortunately. And so scurvy is actually a very big concern in a lot of areas, especially areas that depend on like one of their major crops is like rice. And so one of the ways that people have gone about trying to like reduce scurvy, as well as another condition called vitamin A deficiency, which leads to blindness. A lot of these foods also have vitamin A in them, is to generate food sources like rice or corn or wheat that actually have been genetically engineered to produce vitamin C, so or vitamin A. So if you've ever heard of like golden rice is genetically engineered rice that actually produces vitamin C and vitamin A. So the hope is that if you were to deploy this in areas that are not rich in like fresh foods, or the accessibility to those fresh foods is reduced, that they would still be able to eat their primary crop, which is rice, but they would keep getting these nutrients, which would basically eliminate scurvy and vitamin A blindness worldwide. So why don't we do this? Because did you notice that I just said that we had genetically engineered the rice to express vitamin A and vitamin C? And when I say that we genetically engineered something, some of you may have been like, okay, whatever. Some of you may have been like, ugh, that doesn't sound good. And that's okay. Hopefully we'll get to talk about it later in the semester if we have time, although the provost keeps making that a little challenging. But I want to talk about bioengineering because actually we have the technology for golden rice. It's actually illegal to deport. And the reason it's illegal to deport is because people don't understand what genetic engineering is. So scurvy is still a worldwide issue. Vitamin A blindness is still a worldwide issue. And it's really, I mean, there's a lot of complications involved, but a lot of it comes down to the fact of this huge push against genetic modification. So anyway, hopefully we get to talk about that. I would love to talk about that with you all. But today, we can't talk about it today. But I do want to mention another connective tissue disorder. So again, you can't live without connective tissue or tissues, but there are several disorders characterized by like defective connective tissues. So scurvy is one of them. Another is a genetic disorder called Marfan syndrome. And this is where there's an abnormal gene on chromosome 15, and this leads to, again, just like abnormal connective tissue. But what happens is this leads to kind of like longer fingers and toes. So you can see on the left here, this is the hands of somebody with Marfan syndrome. You get excessive growth of the ribs, and this is because, and the vertebral column. So anywhere where you have like intervertebral discs or a lot of like connective tissue discs. So in the lecture, we talked about like cartilage. Cartilage is connective tissue. Bone is connective tissue. Yeah, blood is connective tissue. A lot of excessive growth of those tissues. Weak tendons and ligaments, because basically your bones and are kind of growing too fast for your ligaments to accommodate, but as well as that, like just the structural integrity of those is weakened. Weak blood vessels, lens fibers, which help kind of hold the lens in place in the eyes, are disrupted, and so you have impacted vision. So the symptoms of Marfan syndrome, as it says, kind of show up around 10, but the vascular issues leading to sort of issues with the integrity of the vascular system starts to cause issues around the age of 50. And so this says mortality, which is sort of like an increase in the potential for death, and occurs around 50 years old. Not to say that people with Marfan syndrome don't live beyond 50. They do, they can, but there are a lot of complications beyond that age. So we're almost out of time. We have like 5 more minutes. I did want to just like really quickly address this question because I've been wanting to address this question repeatedly. Somebody asked me, why can't we drink soap to kill viruses, which I love? And the reason is because in a lecture, we talked about how soap destroys lipids, and which is really nice because, for example, like coronavirus, which is being shown here, has an envelope made of lipids, specifically made of phospholipids, actually, your phospholipids. And that envelope is actually what attaches to cells and allows the virus to get into the cell. So soap eliminating the lipid layer on viruses prevents it from getting into cells. So this is why when you wash your hands, you're killing the virus, you're like eliminating its envelope, which means it can't infect. But also it's like the virus won't be able to do anything if it can't get into the host cell. So you're killing that virus for all intents and purposes. I know it's not alive, but it's like killed. So why can't we drink soap? Like, what if we get infected with the virus? Why can't we just drink soap to kill it? And the answer is twofold. One, if a virus is in your body, it's in your cells. So you can't get the soap into your cells. So the virus, once it gets into your body, it's like in your cells, it's hiding out. This is another reason why antiviral medication is very difficult. It's because it's really difficult. Your cells are very, very selective with what gets in them. Viruses can get in them because they have the lock to your keys. Remember, it's a very specific process. And so getting soap in there is actually more difficult than getting a virus in there. So that's number one, viruses are in your cells, so they're hiding. Number two, soap destroys phospholipid membranes. And remember, viruses their envelope is phospholipid membranes. So, right, phospholipid membrane, so that your body doesn't detect it as bad. This is detected, so you see the virus entering the cell, making more virus assembles. But if you'll notice, this is like a little bud. Virus exit your cells by exocytosis, or like budding. But when this happens, they are wrapped in your cell phospholipids. So that means that a cell like the viral envelope is made of the exact same material as your cells. It's made of, if this is a human virus, the viral envelope is human phospholipids. And so, yes, soap can destroy those phospholipids, which is okay when you're washing your hands, because the skin on your hands, the cells that make up the skin on your hands are dead. So when you're washing your hands, yes, like the soap is disrupting all phospholipid membranes, but your cells on the outer surface of your skin are dead. So it's okay if the soap also like gets rid of some of your outer dead skin cells, right? That's okay. But, oh, sorry, my cat is upset at me for, I don't know, not feeding him every 5 minutes. But the problem is, if you were to drink soap, the cells inside of your body, making up your trachea, making up your stomach, making up your small intestine, those cells are very much alive. And if you were to drink that soap would eliminate any phospholipids, including the phospholipids of your living cells. So soap is toxic. but it's only toxic if it's ingested, right? Or if it encounters living cells. This is also why if you get soap in your eye, like it hurts if you get soap in your eyes too, right? Soap is not to be internalized. Soap is fine on the outside of the body because your skin is made up of dead cells. So it's not impacting anything. But if you drink soap, this is toxic because of the fact that all of the cells inside your body are alive and you're killing those phospholipids. So this is why, oh man, I had a picture of like dead skin and living skin, but whatever. But this is why do not drink soap. This is the same thing. Don't drink or ingest antiseptics or disinfectants because very similarly, you can wash things, but they're okay on dead cells, but not living cells. So if you, oh, sorry. I just want to clarify one thing really quick. This is the difference between something when you hear that it's a topical medication. So like Neosporin is a topical antibiotic. Topical means it's okay on dead skin cells. So topical means on the surface of your skin, whereas some antibiotics are okay if they're ingested. So it's a big difference there. Okay, sorry, JC, what's up?
I'm not the only person who I'm not the only person who had their parents like make soap when they said bad words. How
No.
dangerous is that?
Yeah, I also had to do that. I guess like, you know, we're still here. And my suggestion is just don't do that to the next generation. But I think the thing about it is it would take, I oversimplify because yes, the cells inside your body are living. It takes quite a bit of soap to kill them. So obviously like we're still alive even though we had our mouths washed out with soap. But I'm like, I probably what happened is when you had your mouth washed out with soap, like you immediately spit it in the sink and like got it out of your body. If you were to ingest a decent amount of soap, like a bad amount of soap, what would happen is you would throw up. So your body knows. Your body's like, this doesn't belong in me, get this out of here. And so it throws up. Yeah.
Just curious. That's what it had me thinking about. Because I remember.
Don't say any more bad words ever. That's a good question. Are there any other questions? We're actually out of class time, but I can hang out for a couple more minutes. I do have to take my daughter to get her flu shot today. Yes, yes, yes. We love flu shots. We get flu shot presents. We're excited. We celebrate shots.
My brother is super allergic to the flu shot. It was really weird how we found out because he just got the flu shot because we didn't think anything of it, and then his arm just like swells up while we're still there. He's allergic to
egg allergy. So
Yeah.
you can get some that don't have albumin. They have specific ones without albumin.
He's
Although-
allergic to it. We're nervous, but we
Yeah.
do like we still get it because at this point Colby got the flu last year and he's like, you know what? I'd rather get sick.
Yeah. Well, and like I mentioned, inflammation, a little bit of inflammation is good. Some people are reporting this year that with the flu vaccine, so I don't get the albumin containing one either just because like eggs used to make me a little bit sick. But I got the flu shot this year. I never really have any reaction to it. But I got a little bit of like a rash or light inflammation at the site. But again, it's a controlled, decent amount of inflammation is good. So anytime I have soreness or anything as a result of the vaccine, I'm always like, yes.
I hate needles. Like I have a love hate because I think they're so cool. It's so cool how we like put stuff into our body that way. I think it's so neat to learn about. But also like I requested to get a vaccine that my parents skipped on recently, and then they're like, bring out the needle, and I'm like, I believe in science, so I have to do this. It was
I chose an interesting field because I have a deep-seated, horrible fear of needles. So I'm not really sure. And yeah, I am with you. I don't have a problem like if a needle's like within my view. I have, for some reason, like my weird thing with them is needles going into like veins. I can't handle. I think I might have a thing with veins and not needles. I don't really know. But the whole situation, like I pass out when I give blood. I pass out when I have like IVs. Even talking about it is making me a little nervous. I'm with you.
Blood, but I'm like really tiny and don't really weigh a lot even for my relative size, so I've never been allowed to give blood. So I always the theater department at my old school would always be the ones who ran the blood donation. For some reason it was a very weird choice to have the theater kids give blood, but I always just I helped out, and they're like, you want to donate? And I'm like, if you're okay with me passing out in the chair.
I know. My friends always nominated me to be the chair of the blood drive committee because they thought it was hilarious. And I always passed out. Whatever, I got free orange juice, which I loved.
See, I'm lucky I don't have like a blood aversion because ever since I was a really little kid, and it's weird, like my sister had it too, but I get really chronic nosebleeds.
Oh no.
Yeah, like even this morning I was in the shower and just suddenly like there was blood on my chest and I was just like, yeah, okay, that sounds about right.
Arizona's so dry. I don't know.
Yeah, that's the thing. It's so dry. And
Yeah.
like my mom, apparently she used to have them when she was a kid, but her doctor, I guess it was a different time, I don't know. Her doctor just like cauterized the blood vessels in her nose really quick. And she's never had another nosebleed. And so she took me to an ENT one time to say like, hey, do that. And they were like, we can't do that anymore. And she's like, why not? Because it's not 1960.
Not exactly. Whoa. I would say get a humidifier. That's what I do.
Because humidifier, I got an expensive computer and humidity scares me. So I try to keep it as dry in here as possible.
I'll tell you, my humidifier is, if you can find one, clutch.
Like I went so far, like I have an air compressor, but the nature of compressed air is you get liquid. Sometimes it condenses into water. And so like a lot of people say like, oh, just take the air compressor, like to clean out dust of the PC, right? Because like it's
oh my god, yeah,
like,
I don't know. Yeah,
custom built pretty. And I was like, I don't want to put water on that, so I went out of my way and I bought this thing. Strongest plug-in air blower that you've ever seen, dude.
Whoa.
It's so nice. I just keep it by my desk.
Yeah, I don't even have a computer. I have my laptop and it's like so old. Actually, somebody just gave it to me, so.
Hey, now's the time to do it, dude. Technology keeps getting better and cheaper at the same rate. Like Nvidia just dropped a new line of graphics cards that, like, I thought mine was good. I have a 2070 Super. Their bottom rung new one of the 3080 series blows mine out of the water, like 3 times better. And I was so mad because I just paid like $500 for this thing, and then they're like, "Oh, hey, $399, you can get this one that makes yours look like Garbo," and I'm like, "Sick!"
Yay!
Were you able to do
questions? Oh, sorry.
No, I was just asking if he was able to do the biology lab experiment thing. I can't pull it up. It keeps on telling me there's a JavaScript error, and I'm following all the instructions he gave us, and I don't know what I'm doing. And I'm very worried that maybe just my laptop doesn't allow it.
No, JavaScript error sounds like a driver issue. You might just want to update your drivers for Chrome. Or if you really want to go hardcore, uninstall Chrome completely and then reinstall it, and it should reinstall to the most updated version.
I have a Chromebook. I don't know if I could uninstall Chrome.
You should.
Yeah,
it does need to be the most updated version. So you might see if you're up to date. That's the only thing.
Yeah.
Email me though, so that I know. But I would try checking to make sure it's up to date.
Okay.
It's either that. It's usually the necessity of an updated browser, or that you need to clear your cache or cookies.
I've done that a few times, just like every time it stops around like 71, and then it's like JavaScript error, and I click it, I try to run through the list of everything it says to do. But
I would try to see if you're up to date. But keep me in the loop.
I will.
Sephora, I saw your hand up.
Yeah, this is kind of like a lengthy question, so if you don't have time, you can just email me instead. But I still don't understand like the difference between leukoplast and amyloplast. Like I'm just like stuck on it, I guess.
Oh, that's okay. So that's what I will say first off is you don't need to worry about it. At least not for like moving forward. The big thing is that like amyloplast, so amylo stands for like starch. And so amyloplasts store starch. And then leukoplasts.
I know it's like amino acids, proteins and oil or something.
Yeah, so it's sort of more similar to like another way to get like proteins and lipids, I guess. So like the amylo specifically is like starch, carbohydrates. Does that make sense?
So it's just like all it does is just like store. That's all.
I know. Yeah, basically.
Oh, okay, so it's just storage. Okay, I don't know why I was like thinking so deeply about it. Like I thought it was like had like a function or like something.
Don't worry. I mean, not really. Don't worry too much about it though.
All right, thank you very much.
Yeah. Have a good day. Queenie, did you have a question?
Oh yes, Dr. C. So about the organelles project, so I created a video for mine, but the video is made up of different slides. It's not really a PowerPoint, so would it still count as?
Yeah,
Okay.
that's fine.
Okay, yeah, because I didn't want it to get mixed up with the PowerPoint. It's not a PowerPoint, but it's a video with like different slides though.
That's totally fine. As long as it's just like the information is, presented in a fun, understandable way, I'm good. Don't worry about
Oh, okay. Thank you
Yeah.
so much, Dr. C.
it. No problem.
Okay,
Mhm.
have a
Yeah,
good
then
day.
start working on stuff. I'll catch you
Have a
later.
wonderful day. Bye.